Hi, my name is Dr. Sarah Barnett and I am a recipient of the iMig 2020 Young Investigators Award. I am a postdoctoral researcher in Professor Judy Coulson's lab at the University of Liverpool. My general aim of my research is to find new therapies for mesothelioma. Median survival of mesothelioma is around 1 year, and treatments such as chemotherapy that work for other types of cancer have less benefits to patients with mesothelioma, and surgery is often not a viable option as mesothelioma is usually detected late and so is at a more advanced stage. Therefore, this means that effective treatments for mesothelioma are urgently required. In order to try and find a treatment that works for mesothelioma, we need to understand what changes have occurred in the tumour in comparison to normal, healthy tissue. Our lab is particularly interested in an enzyme called BAP1. The gene that encodes for this protein is mutated in around 50% of mesothelioma cases, resulting in the loss of the expression of this protein. As BAP1 plays a fundamental role in many normal processes in the cells in our body, when BAP1 is lost, then the cancer cells have to adapt in order to survive. We have been focusing on finding out what changes occur as part of this adaptation, in the hope that we can find new drug targets that are specific to BAP1 mutated cancer cells. So far, we have identified 41 proteins that change following BAP1 alteration that are targeted by FDA-approved drugs already approved for the use in humans to treat other diseases. We hope that one or more of these drugs could be repurposed for the treatment of mesothelioma. By focusing on repurposing drugs already in use in the clinic, then any drugs that show promise could make it to clinical trials much more quickly than brand new drugs. Many of the BAP1 dependent targets we have identified control how tumours interact with the surrounding normal tissue and can affect how it spreads locally, known as invasion, or through the body, known as metastasis. A lot of cancer research, in our lab and labs all over the world, is performed on tumour cells grown in plastic dishes. However, this does not really recapitulate the environment that the tumour cells would experience in a patient. The gold standard model to test the behaviour of tumours in a 3D environment within a living organism requires the transplantation of mesothelioma cells into mice, and this is known as a mouse xenograft model. However, mouse models are very expensive to purchase and maintain, and they are also protected, and so any research requires an animal license. And so this leads me to one of the primary objectives of my current research, which is to develop a method that will reduce or replace the use of mouse models in mesothelioma research. We are developing methods to use fertilised hen's eggs to grow mesothelioma tumours. This model is considerably more ethical than mouse models, with the added benefits of being cheaper and much more high throughput. This means that money provided for research into drug development for mesothelioma will go a lot further. In addition to this, it is a particularly good model for testing the effects of drugs on tumour growth, as it is easy to both administer the drugs and monitor the effects of the tumour over time. And so this alternative model will allow faster pre-screening of drugs for the treatment of mesothelioma, providing potential for faster translation from bench to clinic, whilst also reducing and/or replacing the use of mice in mesothelioma research.